Hello, biology period 3 on Green Day. I hope you're having a stellar Friday. I'm right now going to be going over the cellular communication assessment. Now, this looks very similar to your cellular communication document that you've been working on because all of you have been putting in images and maybe labeling those images in this section of your Google Doc, and you've been finding lots of great sources and defining lots of great words to answer how your cell receives or gets a signal, has signal transduction, and then finally has a response. Now, the assessment that I'm going to be grading is partially going to be dependent on your research, but it's now going to be putting your work into one cohesive idea. And to do that, you are going to be putting in one or more labeled images of your cell signaling process, and then describing each step based on 3 simple questions. I don't anticipate that this is going to lead to a lot of new work. You should have most of the answers already, but rather you're going to be thinking about each step in a thoughtful manner that really shows me that you understand how changes occur within a process to make the next step occur. So let's get started. First thing that I need from each of you is to make a drawing of your cell signal. So I am going to be doing insulin with beta cells, surprise, surprise, for my example. So I'm going to go right here and I'm going to go to Insert. I'm going to do Google Drawing. I'm going to do from new. And then I'm just going to Control Paste. Oop, that's not what I wanted. So I'm going to have to do Image. I'm going to have to do by URL. I'm going to paste the image here and we're good to go. This is the image I'm going to be using to describe how pancreatic beta cells make insulin. Now, what you want to do is label each specific step that you want to describe in your assessment. So to do that, I'm just going to draw a box right here, a text box. I'm going to make sure that it is shaded so I can see it. And then I'm just going to do a 1. And the 1 corresponds with glucose going through GLUT2. Now, if there were multiple steps for this basic step that I wanted to be a little bit more detailed on, I could do things like 1.A, and then maybe 1.B. You get the idea. And when you are finished labeling this, and maybe you go back and change your labels as you do your assessment, you will press Save and Close, and it should be good as such. So let me go through the 3 questions you're going to be answering for this assessment. You're going to first put in each step. Now, my first step or question with describing this step is going to be describing what came before this step initiates. Well, all of us are starting off with a cell signal that sort of initiates a response within our cell. Something outside of that cell is causing this signal to be formed. So you really want to think about the change in an environment or another cell or another tissue that's leading to this action. So for insulin production, the change that happened before is an increase in blood glucose levels due to the digestion of carbohydrates in the small intestine. Right? Blood glucose levels are going to rise as a result of us eating food and having carbohydrates being digested in the body. So why is this step happening? This is where you want to think about things such as protein shape and function, ATP energy, changes in environment, or enzymes. Why is this step happening? So I can talk about glucose needs to enter the cell through a transport protein because it is hydrophilic. GLUT2 operates and functions by having a hydrophilic core. And I know that GLUT2 functions and opens when high blood glucose is detected. So these are some things that are talking about protein function, molecular type and specificity, and how the protein operates to make this step doable. What happens if this step doesn't occur? If the GLUT to protein doesn't work, blood glucose levels will stay high and remain in the cell. Cells won't get new sources of energy for cellular respiration. You can think about this last step in a micro and macro level, so this is very much a micro level. If the GLUT2 protein didn't work, if we didn't have this transporter, we would not get glucose into the cell. The macro or the big scale is that if none of this happens, we won't secrete insulin, which means our cells in our body won't be able to absorb glucose. You should be able to use your keywords, your ideas, and your knowledge from these steps to help you walk me through each of these steps. I would say right here that this sort of work is easily a proficient level piece of material in terms of using specific proteins, specific key ideas, and really talking about the cause and effect with each of these. I'm hoping that you're able to finish this for Monday. If you need anything in terms of support or questions, please email me and I will try and get back to you over the weekend. Have a great Friday. Take care.